Hello all, welcome back to the BioRocks channel. Today in this video, I'll be giving you a glance at the whole digestion process that happens from your buccal cavity to that of the small intestine in this one single video. So this will be helping you in preparation for the NEET examinations as well as the boards. So this will be one video where you'll get a glance of all the digestion process. So let's start without any delay. I will be adding such many videos. Please do subscribe and like the video if you liked it, and do please comment in the comment section how it helped you and how you like this video. Thank you all. So now I'm starting the digestion. So the first thing that we would like to see is going to be the digestion in the buccal cavity or the mouth, right? So the first thing that we are going to see is going to be the mouth or the buccal cavity. So here you can see that we are going to see the mechanical digestion, such as like cutting and crushing of the food, mixing of the food with the saliva, right? And we make a bolus, right? So the food is going to be turned into a bolus and it's going to be swallowed. Okay, so these are the ones that you need to understand. And remember that the most important thing to be remembered here about the digestion is going to be the one which is going to be the starch, is going to be the substrate which is being digested by the salivary amylase, right? So the salivary amylase is going to be the enzyme here whose pH is going to be 6.8. Okay, that here it's going to be converted into which form? The starch is going to be converted into a form of maltoses, right? And it can also be converted into dextrin. Okay, so this will be the digestion that you can see here in the mouth as the or the buccal cavity that we are going to talk about. And remember that your salivary amylase, which is also called as tyline, is 6.8, right? And you can even see that the 30% of the starch is digested by the salivary amylase. Okay, it's going to be only the 30% of the starch that is going to be getting converted into the maltoses and dextrin. Okay, and there is no action on the proteins, no action on the lipids. Okay, this is the important thing to be noted here, that no action on proteins and lipids here. Okay, only the starch is being digested, only the 30% of it. So these are not being digested because of the absence of proteolytic and lipolytic enzymes here. And the mucus of the saliva is going to make the food into bolus, right? Now the bolus enters where? Into the stomach. Okay, and in the stomach you can see that your food is going to be mixed with what? So let me write it here for you, that mucus along with the food is converted into bolus, and this bolus will be swallowed, right? This is going to be done. And next one that we are seeing is going to be digestion in your stomach. Okay, in the esophagus, that is in your food pipe, there is no action. It's only peristaltic movements that pushes your bolus into the stomach, right? Now in the stomach, what happens is you can see the presence of these gastric glands, right? So here you can see the presence of the gastric glands, and these gastric glands are secreting the gastric juices. They are secreting the gastric juices here, and the food is going to be mixed with the gastric juice. Actually, the gastric juice is nothing but what? It has a composition of what? Let me write this for you. So this is going to comprise the mucus. It has hydrochloric acid. It has prorenin, right? It has pepsinogen, okay, and it has gastric lipase. So these are all the compositions of the gastric juice in whole togetherly. If you're going to look at this, the mucus, the hydrochloric acid, the prorenin, as well as you can see the pepsinogen, gastric lipase. These are all the things that you can see. Even in the stomach, you can see the mechanical digestion, which is nothing but the turning of the food, okay, where the food is being broken down into more smaller, simpler pieces, and chemically also they are being digested by these all enzymes which are being present, all the enzymes like these, like prorenin, the pepsinogen, the gastric lipase, all the enzymes. Now, coming on to these ones here, we'll directly see the action of these all ones. So if you're going to look at the mucus, this is going to lubricate, right? Mucus is going to lubricate and it is going to protect the gastric mucosa from the action of this acidic hydrochloric acid. And talking about the hydrochloric acid, it is going to kill the bacteria, and it is stopping the action of the saliva, and it softens the food too. And remember, children, that hydrochloric acid is going to convert the pepsinogen, the inactive form of enzyme which digests the proteins, which is a proteolytic enzyme. The pepsinogen is going to be converted into an active form, which is being called as pepsin. So this is an active enzyme, and this is going to be an inactive enzyme. Okay, these are, that is, the inactive pepsinogen is converted into active pepsin by the action of the hydrochloric acid here, that is there. And remember that even your prorenin, okay, even the prorenin, okay, let me correct the spelling, prorenin enzyme, okay, this is also going to be, these are usually present in the fats, okay, these are present in the fats, and especially the milk fat. So these are also converted by the hydrochloric acid into an active form, which is being called as renin. So this is an active form, and even hydrochloric acid is killing the bacteria, right, that you can see. And next, coming on to when the pepsinogen is converted into the pepsin now, and these pepsin is going to act on the protein. So you can see that proteins are being digested by the action of the enzyme pepsin, into which form, into another form which is being called as proteoses and peptones. It's been converted into these forms. And remember that the one which is being called as renin now is going to act on the casein, the milk protein, okay, the milk fat, okay. Casein is being converted by your renin, okay, renin. That is converted into which form? It's going to be converted into para-casein. Let me take the color. So it's been converted into para-casein, okay, as well as remember that this is going to release out the whey proteins from the milk, whey proteins from the milk, as well as calcium also. Okay, these are important to be remembered. In adults, renin secretion stops. Okay, in adults, this is important. In adults, renin secretion, renin secretion stops. Okay, but yet we are going to have curdling of milk. Curdling of milk is going to be seen yet in the adult intestine, that is especially your stomach part where I'm talking about. Now, coming on to the lipase, what that is going to do is your gastric lipases, which are being present in the gastric juices. These are going to be actually, they are weak in nature. Okay, this is important for us to remember that the the gastric lipase is weak in nature. Okay, it's weak in nature, this point. And remember that there are no emulsifiers. There are no emulsifiers like bile juice here in the stomach. Okay, no emulsifiers in the stomach, due to which we see that inactivated, that is, the fat digestion is inactivated here. Fat digestion is inactivated. It's been inactivated here in the stomach, okay. And this is one important thing. And another important thing is there are no amylase found here. There are no amylases or carbohydrolases which digest your hydrolases which digest carbohydrates. If you can see, I have written only the prorenin, renin, the pepsinogen, the gastric lipases, but there are no such enzymes to digest your carbohydrates here. Okay, that is seen in the stomach here. And next, coming on to the digestion in the small intestine now. So when I talk about the digestion in small intestine, digestion in small intestine, I would like to request you children to just please prepare a tabular form of this way of video. If you make the tabular form and just fix it nearby your study room, this will be helping you in revising very well rather than sitting and spending hours together. If you can just have a glance at the tabular form every day, so that is going to be a better revision for you all. Now, coming on to the digestion in the small intestine, you can see that there are three different types of juices here where the food is being mixed, children. And this one is going to be actually the first thing that you can see here is going to be the bile juice, whose pH is going to be 8. You can even see the presence of the pancreatic juice, whose pH is going to be 8.8. And at last, you can even see the presence of the intestinal juice with their own respective enzymes. And the pH is going to be 8.3. Okay, all are alkaline, right? They are above 7. They are alkaline. So these are the ones present here. But when we speak about the role now, coming on to the bile juice, the first thing that we would like to see is going to be the bile juice, what are present here and what does it do. So to remember this one, actually bile juice is going to be the bile pigments, the water and the sodium carbonates, okay? This is going to have water, it is going to have the sodium carbonates, and it is going to have the bile pigments here. Whole together is going to constitute to form a bile juice. And as we have seen that its pH is 8, it is alkaline in nature. And usually the bile salts which are being present, let me talk about these now. We have the bile salts, and these bile salts which are present here, like your sodium glycolate, I'll write it for you. These are being called as sodium glycocholate. And next we have is sodium taurocolate, taurocolate. Okay, so the bile salts with the help of there is monoglycerides actually. So bile salts and these are going to actually help in emulsifying the fats. Okay, so these are trying to emulsify the fats. What is emulsifying fats is nothing but breaking the large fat molecules, breaking the large fat molecules into smaller particles is being called as smaller fat molecules, is being called as emulsification. Remember, along with the bile salts, it's going to be few of the monoglycerides also, okay? These all, okay? Even monoglycerides are also helping the bile salts to perform this emulsification of fats here. And next, coming on to the pancreatic juice now. What is that doing here? What are the enzymes present here? In the pancreatic juice, you can see that there is presence of pancreatic amylase. Let me first write down the ones here. So it is going to act on starch. The substrate is starch. And the one that you can see here is going to be pancreatic amylase. So we are having the learning about the first enzyme here, that is going to be a pancreatic amylase. We have seen 30% starch which has got digested in the oral cavity by the ptyalin or the salivary amylase. Now again we are getting the pancreatic amylase which is digesting the starch into the maltoses. maltoses and dextrin. Okay, it's getting converted into this. And next you can see that there is an enzyme which is being called as trypsinogen. This is an again inactive form, right? It's a gen form. And this trypsinogen is going to be converted into active form again by one of the enzymes, and that is going to be the enterokinase enzyme, which is an enzyme present in the pancreatic juice, is converting your trypsinogen into an active form called as trypsin. And this is going to be again releasing few of the dipeptides also, but we are not considering them. Just consider this. That is going to be an active form, the trypsin. And remember that this trypsin is going to act on your proteoses and peptones. Okay, a while we have back we have seen about the proteoses and peptones being produced by whom. It was being produced by the pepsin by acting on the proteins. Right now the trypsin is going to act on those proteoses and peptones. Okay, and they are going to convert them into peptides now. Peptides. And next we have another inactive enzyme which is being called as chymotrypsinogen, which is also an inactive enzyme in the pancreatic juice, which will be converted into active form by the presence of this enzyme. Again, the activated trypsin is converting it into an active form, which is being called as chymotrypsin. Okay, this is there. Now, coming on to the chymotrypsin, children, the ones here, the chymotrypsin is going to hydrolyze the peptones. Again, some of the proteoses and peptones which are coming from the stomach here, when pass into the small intestine, the chymotrypsin here, the chymotrypsin is going to act on them, and it is going to produce again peptides, same as trypsin. Okay, and even we are going to have that these peptides are going to be converted into dipeptides and amino acids. Again, dipeptides and amino acids, that is being done by another enzyme here, and that enzyme is being called as, we're going to call it as carboxy peptidases. Okay, so here the carboxy peptidases are the ones which are going to act on these peptides and convert them into the dipeptides and amino acids. But remember, children, I should speak about the pro-carboxy. They are not present in active form. They are present as pro-carboxy peptidases. Okay, as this they are present in the pancreatic juice. But the pro-carboxy peptidases will be converted into carboxy peptidases again by the action of your enzyme, active enzyme called as trypsin. Okay, this is the one which need to be remembered by you all. So that is about it. And next, coming on to the fat digestion, children, here we are going to have pancreatic lipases. So the emulsified fats coming from here. So we have seen that the bile salts along with the monoglycerides have emulsified the fats. Now these emulsified fats will be acted upon by the pancreatic pancreatic lipases. Okay, so this is the enzyme now which acts on the uh, these emulsified fats, and they are now converting them into the fatty acids and glycerols and monoglycerides. Okay, and next one, you can even see that even your there are some of the enzymes like your ribonucleases. Let me write this for you. The ribonucleases are there, and we can even see deoxyribonucleases. Okay, so these are the ones which are going to act on what? They are going to act on your RNA present in the food into ribonucleotides. Okay, into the ribonucleotides respectively. And the DNA will be converted into the deoxyribonucleotides. Deoxyribonucleotides. Okay, this is a one whole togetherly that is being seen by the bile juice and the pancreatic juice. Then coming on to the intestinal juice now. Okay, what does the intestinal juice do and what are the enzymes present here? That we are going to see now. Now, coming on to the ones here, remember that the invertase enzyme, okay, especially starch. Starch is being acted upon by invertase invertase amylase. Okay, this is going to convert the starch into maltoses. Okay, now maltoses will be acted upon maltase, and that will be converted into two molecules of glucoses. Okay, and the isomaltose, because here along with this we can even see the production of isomaltose. So now the isomaltoses will be converted into glucoses again by isomaltase enzyme, which converts that into the two glucoses here. And we can even see the presence of dextrinases also, which convert the dextrins into glucoses. We have sucroses, which are being converted into the glucose and fructose glucose and fructose. This will be done by sucrases. See a whole lot of carbohydrate digestion happening in the small intestine. And the lactose sugar will be converted by the lactases into glucose and galactoses. Okay, glucose and galactoses. And remember that the text talking about these all are about the carbohydrate digestion. This is about the carbohydrate digestion by the intestinal juice enzymes. Now, when we speak about the protein digestion in the small intestine, now remember that it's going to be the amino peptidases which are acting on the peptides. Okay, this is going to be acted upon by amino peptidases. You can even call this as eripsin. If you find it as eripsin, this is the same thing. This is going to convert it into the dipeptides, as well as it is going to even produce a simpler amino acids. And again, the dipeptides will be acted upon by the dipeptidases, dipeptidases, and will be converted into the simpler amino acids. You can see a whole range of enzymes present in the intestinal juice, acting both on the proteins and the carbohydrates. Later, you can even see that they have intestinal lipases, so they are acting actually on the different triglycerides, the diglycerides, okay, and monoglycerides, all these fats whichever are being present in the food now we are going to be acted upon by the intestinal lipase intestinal lipase, and now these are going to be converted into the simpler fatty acids as well as the glycerol. Okay, this is going to be the one. And even you can see the presence of the nucleic acids are also being again digested here, that is present in the intestinal juice. We have seen the same in the pancreatic juices, right? But here you can see that we are having nucleases which are acting on these and convert them into the respective nucleotides. Okay, we are going to have the nucleotides getting converted into the nucleosides and phosphoric acid again, nucleosides and the phosphoric acid, the H3PO4. It's converted into these by the presence of an enzyme which is being called as nucleophosphatases. Okay, nucleophosphatases. These are the ones. And next we are going to have another last one which is going to be the nucleosides. Okay, the nitrogen base and the sugars, they are converted into the free nitrogen bases like purines and pyrimidines and pentose sugars. Okay, these by the action of an enzyme which is being called as nucleosidases. Nucleosidases. So they will be converted into what? the purines, they will be converted into pyrimidines, and they are going to be converted into the pentose sugar. Okay, these are the ones that we can see that the enzymes present and the digestion that is going to happen in the small intestine children. So by this, we are done with the whole digestion process. If you can look back, the type of enzymes present here, so we could see salivary amylase here. Only starch is being digested here. Next, coming into the stomach, you can see actively protein digestion is being done, right? And you can even see the there is no action on the fats, no action on the what we call the carbohydrates. Only the proteins are actively digested in the stomach. Coming into the small intestine, by the action of the bile juice, the fats are getting emulsified. By the action of the pancreatic juice, starch is being digested here, the proteins are digested here, and even you can see that for the first time the fats are being digested here, and the nucleotides, that is RNA and DNA, is being digested. And lately coming on to the intestinal juice here, it is going to do the carbohydrate digestion, the protein digestion, as well as it's going to even perform the fat digestion and work on all the nucleotides and sites here, and the whole digestion is being done. So remember, the key points to remember here is going to be mouth is carrying forward only starch digestion, stomach is doing the protein digestion, and the small intestine is the one where pancreas is going to do the digestion of the starch, the fats, and the proteins, and the liver is doing only the fat digestion. The intestine is doing whole togetherly the complete digestion is going to be done. So children, when I'm going to meet you in the next video, I'll be speaking about the all the hormonal control of the digestive secretions and their actions. I hope this video is going to help you in making out your own tabular form notes for the revision process for the NEET. So if you like this video, please do hit the like button, and please do comment in the comment section how you like this video. Thank you all. Thank you.